Hi friends, you are back with me, Professor Girish Kukreja, and like in last few series, we have been talking about different spot tests or qualitative tests which are used for estimating reducing sugar. Like in our earlier video, we talked about the Benedict's test, right? Another particular test, like in my earlier video, we have also talked about the picric acid test. So there are different tests which can be used to estimate the reducing sugar. I have also defined what is reducing sugar in my earlier videos. I told that a sugar which has a free aldehydic or a ketonic group, which you generally get in monosaccharides under alkaline condition, and then they can act as the reducing agent and themselves can get oxidized. Such particular sugars we call it reducing sugar. And if you remember, I had told that all monosaccharides they are reducing, and yes, there are few disaccharides which are reducing, there are few disaccharides which are non-reducing. In our earlier video, I had asked you that which are the disaccharides which are reducing and which are disaccharides which are non-reducing. I'll tell you an hint, like sucrose, the table sugar which we have, it is a non-reducing disaccharide, and maltose is a reducing disaccharide. Yes, so I hope you did not watch this video before and that video later. So talking about the Fehlings test, right, one of the tests which is used to differentiate between reducing and non-reducing sugar. I'll be focusing on the use of these tests for differentiating between reducing and non-reducing. Fehlings test is also like used to differentiate between aldoses and ketoses. But then actually what happens is your ketose is also under alkaline condition, they get converted into aldoses and eventually they also give this particular test positive. So I'll be focusing only on a qualitative or a spot test of using this Fehlings test for determining the reducing sugar, like whether it is present or not, and I'll not be again focusing on the quantitative part of it. We'll be only talking about the qualitative, whether yes or no. Yeah. So, Fehlings test, as I told, differentiates between reducing and non-reducing. Yeah. So, formation of a yellow-red precipitate similar to what we saw in the Benedict's test, right. So, similar kind of a reaction which takes place here. There we saw in the Benedict's test there was sodium citrate which acted as the chelating agent. Here you have the Rochelle salt, which we call as the sodium potassium tartarate. Another name for sodium potassium tartarate is Rochelle salt, which acts as the chelating agent. To prepare this particular reagent, you will take 35 grams of copper sulfate pentahydrate in 500 ml of your water, 120 grams of your KOH and 173 grams of sodium potassium tartarate in around 500 ml of water, and you will take equal volumes of both, and then you will prepare what is called as your Fehlings reagent. So remember, we are going to use this freshly prepared Fehlings reagent for the test, right? Don't use A separately or B separately. We will be like using both, once you mix these two, after mixing whatever you prepare, that is your Fehlings reagent. So take this Fehlings reagent, and how I am going to do the test is, I am going to take 1 ml of my Fehlings reagent, I am going to add 1 ml of the test solution, and as I boiled this mixture in Benedict's test, I will boil this in the boiling water bath, and again formation of a brick red kind of a precipitate. Here you can see a clear formation of a brick red precipitate. It indicates that the test is positive. If even after boiling there is no change in the color, like here you can see, even after boiling there is no change in the color, we can say that the test is what is called as your negative. There is no formation of a precipitate here, you can see. So that indicates absence of a reducing sugar. What actually happens, you can see that copper sulfate with potassium hydroxide forms this cupric hydroxide, which forms this cupric oxide in the presence of this glucose. So this is your glucose here. So yes, this is say D-glucose, which acts as the reducing agent, and as I told, the reducing agent itself gets oxidized. So your glucose gets converted to gluconic acid. So there is formation of D-gluconic acid, and this particular gluconic acid, and here you have formation of what is called as your cuprous oxide. And it is this cuprous oxide which forms a brick red kind of a precipitate, indicating that your sample, it contains a reducing sugar. Yeah. So Fehlings test distinguishes between reducing and non-reducing sugars. Rochelle salt acts as the chelating agent. You mix both of them, 1 ml of your test and 1 ml of your Fehlings reagent. Boil it in the water bath. A brick red round precipitate, a test is positive. So it's that simple. So stay tuned with me, Professor Girish Kukreja, for more in microbiology, biochemistry, molecular biology, and much more. Thank you so much.